All right. So I'll get through this relatively quickly. So I'm going to talk about the sort of development process that Chip just mentioned that got us to FDA approval of Kymriah. And Kymriah is cystogen-leclocel, which is actually more pronounceable than the name for Zolgensma. Does anybody work with it who can actually say the name?
Zolgensma?
Yeah. No, Zolgensma's easy. The generic.
Oh, no. No,
Yeah.
Nobody bothers.
And I don't know how you pronounce XIOI, which is at the end of it. So at least by that standard, cystogen-leclocel, which is our generic name, and see how they blocked out Kymriah because I'm not allowed to use that. But this is the product. This is genetically engineered T cells. So we collected T cells from a patient. This is just an example. It's not a real one. It says John Doe right there. But what we do is we collect T cells from a patient. We genetically modify them using a lentivirus, which is a modified form of the HIV virus that has no ability to cause disease, but has the ability to modify T cell genomes. And then we end up with a bag of cells that is essentially nearly 100% T cells and somewhere around 15 to 40% CAR T cells, which are the genetically modified T cells that actually provide the therapeutic effect. This is a autologous manufacturing process, which means that you have to collect cells from every single patient. You have to manufacture the cells for those patients. You have to fully qualify that as there were a lot of 10,000 vials of an antibiotic. You have to do all the same release testing for every single patient, and then you have to give it to the patient. But there's no tolerance for release testing that's unsuccessful because we still want to treat the patient, because if we don't, what will happen? Oh, they'll die. So that's not a lot of fun for the drug companies. They're starting with a feedstock that comes from human beings. That's hard. And then they have to make these cells. And then they have these doctors who scream at them if the process doesn't work. And there are often circumstances, actually, where this product doesn't conform. But it's still perfectly safe and probably efficacious. So won't get into that anymore because we're talking about the development process. I'm not going to talk about this. It's just these are the pieces of a CAR, a chimeric antigen receptor. One that binds to the actual tumor cell up here, T cell down here, and then all these parts which actually activate the T cell fully enough to cause both the T cell activation and proliferation. And both of those are required for the therapeutic effect. It started out as CART19. That was first name CTL019. Then the cystogen-leclocel, shortened to Tisacel, and Kymriah is actually the trade name for this stuff. Somewhere around 15 to 20,000 people in the world have gotten an engineered CAR T cell product. This starts, I'm not going to get into this, starts in the late 80s. And the only point here is that pretty soon, like in the 90s, people are doing clinical trials and nothing works, and then nothing works, and then nothing works, and then something works. And so that was the next step. And actually, this is a movie, but I don't think it's going to work. Oh, that's a good sign. All right, start that. All right, watch what happens here. So this is a cancer cell. That's a T cell. That bubbling is the destruction of the T cell. And the important thing to understand about this process is that the T cell doesn't care if this patient has relapsed 8 times. The T cell doesn't care if this patient had a transplant. The T cell doesn't care about the internal genomics of that cancer cell. It doesn't care how high risk that patient is. It kills it anyway. And so in a recent paper we just published in JCO last year, we saw 97% of our patients go into remission. So this, again, quickly, starts with a clinical trial in adults, the way it was intended. So three adults treated by the group at Penn, David Porter, with CLL, chronic lymphocytic leukemia, which we never see in pediatrics. Those 3 adults, so that's all it takes. What the FDA, not the FDA, well, the FDA too, but the IRB is really what I felt like I was dealing with. What the IRB and the FDA want is some evidence for potential benefit. You cannot give an experimental agent to a child without an evidence of potential benefit, or rather, it's very difficult to do that. Now, the argument for potential benefit is either easy or hard, but it worked in 2 out of the 3 patients is plenty good enough. So that was, the FDA was happy. The IRB was not so happy. It took me a year to get this trial open. The first response from the IRB was 76 stipulations for which my response was 25 pages long. So it was a bit of an effort. But we did it, and we treated a couple of patients, and they both went into remission despite refractory disease with ALL, the most common childhood cancer. So that started the whole ball rolling. And then we 18 months later, pointed out that these patients not only were going into remission, but some of them were staying there. So a 90% complete response rate in adults and kids, including adults over at Penn, and patients who were sustained. And actually, our first patient ever treated, Emily Whitehead, was reported here and then again reported here. And so these are patients who, in some instances, and about 80% of our patients don't go to transplant after CAR T, who are staying in remission after CAR T for longer periods of time. And so this was like, maybe this is definitive therapy for some patients, not for everybody. Then lymphoma, I won't get into that. Adult lymphoma, Steve Schuster at Penn and Stephan at MD Anderson. This is a Kite product. This is the same Novartis product that ends up going forward. And then our, as Chip mentioned, registration trial for pediatric ALL. It is ridiculously uncommon for one of these things to be FDA approved in kids first. And this is a whole new field of medicine. Nothing like this had ever been approved, genetically engineered, and for it to be in kids first is unheard of. And it was a fairly simple reason why it worked out that way. We made it work and no one else did. This is a whole chop thing, even though this was an international trial. So our first patient treated in 2012, name known to many of you, Emily Whitehead. The global registration trial that Chip mentioned, Eliana, starts in 2015. This was 25 centers in 11 countries. Canada, US, okay, that's close. EU, that's across one ocean. Japan and Australia, that's across another ocean. And there was actually a fairly simple device that was probably the most important thing in that trial. And that was this, my cell phone, because I was on call, as my wife will tell you, 24/7 for 2.5 years straight to the entire planet. So that was fun. Not doing that again. This then went to the Oncology Drug Advisory Committee, and I'll talk to you very briefly about that, and resulted in FDA approval. So first pediatric patient, 2012. The reluctant realization by Novartis and other people that we were actually getting it to work in kids before they were getting it to work in adults, and they got it to work in adults. I want to be clear about that, but not right away. So it's like, oh, darn it, we got to do this in kids, and they did. So first FDA approval 5 years after we treated the first kid. That's not bad. This was the first CAR T. This was actually the first gene therapy ever approved in the US. And now, and you're going to hear about Luxturna, which is an amazing story. 2022, we have 6 CAR Ts, all for blood cancers, by the way, and then 2 infused gene therapies, Luxturna and Zolgensma. That's what we got. And by the way, does Zolgensma cost?
Is that the 225,000 dose?
No, that would be
That's the other one. That's $1 million.
$2.1 million. That's what it costs. This stuff costs $475,000, in case you want to know. So we've hit the 10-year mark with this young lady, seen on the Today Show this morning, Emily Whitehead, who got into remission with CAR T, has remained in remission with CAR T, and actually never got the bone marrow transplant that she came to CHOP for. So this is kind of a cool outcome. So she got her cells there, and there they are here in her body, and she's still in remission. So lots of argument about whether this is a cure or not. I think you get to a decade, that might be a cure. Timeline, so stretch it out a little bit. Bench lab work was in 2007. Now, I'm going to estimate the percentage of that, and I hate to say this, Chip, because you kill me when I say it. The percentage of the bench work that was presented as IND-enabling experiments to the FDA that was GLP, and that was 0. Not a single one of them.
Always exempt.
Yeah. And it ought to be GLP. And it's great that they're supplying this setup so that they can be GLP. And it gets the whole issue out of the way. But the more innovative it is, the more potentially exciting it is, the more wiggle room there is. A Me Too drug that's 14th in its class, the next statin, ain't never going to happen. This, maybe it's going to work. But, you know, the reality is that GLP would be better. But the other reality, I don't know if anybody's actually in a lab here. Okay, so none of the labs in shop for GLP, basically. You know, mine isn't. I've been here for 25 years. Nowhere close. Because those have regulatory implications that we don't meet. and would take a lot of staff actually to do that. And I'm not saying it's the right thing to do by any means, but there is some wiggle room. I got IND approval, multiple New England Journal articles, breakthrough status, and then the FDA approval. So we started out in 2007 with some of the initial IND enabling experiments. And so there's a 10-year span to FDA approval, and then clinically a 5-year span to FDA approval. So not bad, not bad. And this was the Eliana trial. I already described this. I don't need to go through this. I think the only thing to point out is that we made the cells here in, New Jersey, but that's where they made them. And they went everywhere. So in order to do this, you have to collect the cells here, send them across the ocean in a liquid nitrogen container here, and then send them back frozen again. So it's kind of amazing. But the reality is we had already set this whole process up for bone marrow transplantation. So we actually knew how to do it. And there was both paperwork and logistics. So this is what you heard about all throughout COVID. This was the original cold chain, having to move things at liquid nitrogen temperatures to make this work. So this then, this is the step before what Chip described. So this is a FDA panel that reviews data. And in the oncology world, it's called an ODAC, the Oncology Drug Advisory Committee. It's ad hoc often, but there are some often members that stay on it. And not every drug needs an ODAC or an advisory committee, but many do. And many, this was the first one, so it absolutely required one. So they made us do this. It was many months of preparation, 500 backup slides that Novartis prepared. I did the clinical perspective. And the interesting thing is it's a one-day process, and it's not what you're used to if you're actually used to FDA processes, which is you present all day. There's actually a moment where there's public comment, and there were 3 patient families who commented, Emily Whitehead's dad, and then Emily came up, and then everybody cried. Another patient who had a successful outcome, and then actually a mom who spoke very movingly about the fact that her child had a response. It was actually at our hospital, but it wasn't long enough lived, and the patient actually ended up passing away. And she said this was a therapy that we wished we had access to earlier. And that was, I think, an also important thing for the FDA to hear. At the end of the day, after all this discussion and all the questions, all the squirming, all the hoping that you could answer things correctly, what they do actually is they sit down and they vote in front of your face. So it's not like you get a report 2 months later. I didn't know this. It's ridiculous. And then they show you a slide, and this was our slide. So a unanimous recommendation for approval. Now, the FDA doesn't have to take that recommendation, but if there's a recommendation for approval, it's easy for them. If there's a recommendation against approval, they can ignore that and approve anyway. And the Alzheimer's drug is a great example of that. And that was, I think, not a good idea, but that's what they did. Okay, so then with that recommendation, this is June, then August 30th, 2017, this is actually FDA approved. So you don't know when that's going to happen. Unlike the request for an IND, which is they can just not get back to you in 30 days and you can proceed, although that is happening less and less now. In this situation, it's clearly you can't go forward without a yes, and it can take as long as they want, although there are some constraints on that, and I'm not going to get into all that. And this just, I like to tell this story, came on August 30th, and what I was actually, you don't know when it's going to happen, I was engaged in moving my daughter into her dorm room at Bryn Mawr, actually, at that moment. And all of a sudden my phone blows up and I'm getting texts and phone calls and like, Evie's, what's going on? Is the world ending? And I said, no, the stuff that Dad's been working on has been FDA approved. And she said, great, but that suitcase not moving itself. Back to work I went, but it was still super exciting. At this point now, 6 products are FDA approved. Starting with the pediatric product, and there's Emily across the age range, but you saw her as she looked this morning. And then starting with Kymriah for ALL, Kymriah for lymphoma, Yescarta, which is made by a different company for DLBCL, large B-cell lymphoma, and another disease called mantle cell lymphoma, we don't see very much of. Now, yet another drug for that. All of these recognize CD19 for B-cell malignancies, but now here we have something that recognizes a different target, our first non-CD19 target. Very exciting, simply because, you could argue maybe only CD19 works. Now, it's not just CAR T cells for breakfast. There's other things we can do. And so this is now gene therapy for patients with beta thalassemia, or a disease you're probably more familiar with, which is sickle cell disease. Same treatment. And this is the Bluebird product, and Alexis Thompson is the first author here, and now she's the heme division chief, successfully recruited to CHOP, which we're excited about. And this is an autologous product where you're not collecting T cells, but you're collecting stem cells to correct a defect by transplanting the patient instead of with bone marrow from another person, which has significant risks, with bone marrow from yourself, which is way less risky, and hopefully correcting the defect, and that actually works. This is a gene addition process, just like the CAR T cells are. So they use a lentiviral vector to actually add the gene in and fix the problem, which is great. And if you don't like gene addition, then you can try gene editing using CRISPR. And so this is now in the process of getting toward FDA approval. Data was reported at ASH, the American Society of Hematology, and then in the New England Journal of Medicine at the same time. And that's going to be updated data. And both of these products, the Bluebird product and the Vertex product, and I run this study steering committee, so full disclosure on that, look like they're heading toward potential FDA approval in the next 18 to 24 months. So we'll see if that actually happens or not. And then that would be the first engineered stem cell approach. And Scott Gottlieb, who is the head of the FDA, says that this is something that we ought to see 10 plus products a year by 2025. So it's blowing up, and these things are darn expensive. So what are our issues? Cost. Oh my God, these are expensive. Some of this is because they're ultra orphan indications like spinal muscular atrophy. And then, kind of the insurance world doesn't care that it costs $2 million because there are not that many of those kids. But as you get into, sickle cell patients, that's maybe 1 to 200,000 patients who could potentially benefit from this therapy. That's a whole different deal. What are you going to do about that? Manufacturing. So these are cells. So Zolgensma is a virus that you infuse into the patient. These are cells that you modify the cells and give them back to the patient. So this is really, really complicated, but we're making it less complicated. And then this issue of how do you pay for this stuff. So this is the Kymriah price. Everybody freaked out when this stuff was FDA approved. It was almost $500,000, and $475 is what they came up with because it wasn't $5. I guarantee. I don't actually know. And at this point, I'm indelibly associated with the highest single cost therapeutic in American medical history. Yay! But now it's like just amusing because it's a quarter of what Zolgensma costs. And this, the sickle cell stuff is probably going to be somewhere between 1.5 and 2.1 million dollars is probably what this stuff is going to come at, because these are the ranges that we have. However, what we really need here, so amusingly, the stock ticker for the company that makes Luxturna, their stock symbol was, before they were bought, once. And the idea is this is curative therapy, and that's why it's worth this much money. But there has to be a way at some point to pay over time, because, yeah, it worked for a year, awesome. Works for 2 years, awesome. Now I can't see anymore, or my leukemia comes back, not so awesome, or my sickle cell disease, I'm starting to have a crisis again. And then there needs to be a way to sort of stop paying because the value to the patient no longer exists. So all the companies talk about this. There is no mechanism whatsoever. None. They all talk about it, but how do you do this? Why, people change insurances 5 times. Why would the second insurer continue to pay for something that the first insurer did? No clear answer to that. But this is an issue for these high-cost therapeutics that are incredibly important. So I've really focused not on the sort of initial part of the process the way Chip has, but on the clinical part of the process, which is all sort of brand new, was brand new to me when we started working on this back in about 2013, 2014. We've learned a lot along the way. And the other thing that CHOP did in it, so number one, got it FDA approved because we did the best job with the kids. Number two, learned how to control toxicity, which is considerable with our first patient with Emily Whitehead. I didn't even talk about that. Number three, treated the first commercial patient, because I can tell you, as the guy running the trial and the guy having done all this work, that I was going to die before I allowed any other hospital to give the first commercial product. And so this was FDA approved in August, and we treated a patient in September. Not bad for something that takes 22 days to make. So that's another aspect of this. So commercialization is something that we're all learning about, and I think we're doing better and better.